And welcome, Freddie, and over to you.
Okay, thanks very much. Thanks, Yadvina, for the invite and for the introduction. So I'll just try and share my screen. Works. So I'll assume that everything is okay and people can hear me. If not, please let me know. Hold on one second. Let's see if I can. Yeah. Okay, so I'd like to talk today about Amazonian tree dominance across forest strata. And then I'd start just by introducing why then I think tropical plant diversity is important. So about 70% of global biodiversity is concentrated in the tropics, and then much of that is in forest biomes. And this diversity then really underpins globally relevant biogeochemical cycling. So about 30% of the global carbon sink is accounted for by tropical forests, by tropical plants, trees. But we're losing these forests at a rate of about 10 million hectares per year. So yeah, tropical forests hold a really important role, and for me that's really underpinned by plant diversity, by tree diversity. But measuring this is a significant challenge. So I work principally in Amazonia, and it's a difficult place to work. So and to measure this diversity, so it's huge. It's about 6 million square kilometers. Much of this area is remote and inaccessible, and it's also hyper diverse. So we think there's at least 16,000 species of tree above 10 centimeters in size. So super diverse, really big, and hard to get there. So I'm interested in how we can try and simplify this complexity of diversity. So that's, I guess, one of the primary questions then that drives some of the thinking here. And in that context, for me, a really important kind of idea and paper. So this is based on a paper that was published by Nigel Pittman in 2001 in Ecology, where he made this point that in forests in the southwest Amazon, and then forests much further north in the Ecuadorian Amazon, so this is kind of northwest, western Amazon, although they're separated by thousands of kilometers, there was a small group of species that were really common. And he termed these oligarchs. So these oligarchs, these super common trees, were dominant at large scales. And whilst previously people had mainly been thinking about the diversity of Amazonian forests, how complicated they were, how there was rapid turnover. You can get 300 species in a hectare, and then you go to another hectare and you can get, most of those species will be gone, even though it's very close by. But actually, by setting up lots of field plots, these are 1 hectare plots, he made this very strong point that yeah, actually there's common species there, and they account for a significant number of individuals. And then various people kind of carried these ideas forward, but in 2013, Hans de Stieg published this paper in Science, where he used this very large network of these 1 hectare plots, looking at all trees bigger than 10 centimeters in diameter. So there's over 1,000 plots. I think Hans presented some of this work in this seminar a few weeks ago. And using this really massive data set from across Amazonia, he showed a very similar kind of pattern, which he termed hyperdominance, which is the idea that in the super diverse system, where you might have 16,000 species estimate, you have 5,000 documented. So this is a rank abundance distribution, sorry, with the most common species in the far left, moving steadily towards the least common. But just 227 species made up 50% of all individual stems. So although you've got this really complicated, hyper-diverse forests, the whole of the Amazon, just 227 account for half of all individual trees. So for me, there's really huge value in this framework. So you can understand half of the forest almost at the species level by concentrating on these really dominant tree species. And then if this is just some data then from the data set that I'm going to be talking about later on today. But it makes this point, I think, more clearly that if we just don't use any log axes and we just plot this, again, the same rank abundance distribution from the most common to the least common, we see that there's a few very, very common species, but most species in tropical forests are really rare. And in this data set that I'll present today, about half of all species are represented by fewer than 10 individual species. And almost a quarter, we only have one record. So there's this general point that most species are extremely rare and a few are extremely common. So then some other colleagues, Sophie Fawcett, published this paper in 2015, where they showed, okay, not only are these hyperdominant species really, really common, but they're also driving these important biogeochemical cycling. So they showed that this very small fraction of species is accounting for most of the biomass and the productivity, so the carbon cycling of Amazonian forests. So they have this really, really important role in driving ecosystem functioning. And then more recently, I led a paper in Loreto in northeastern Peru with many colleagues. The three of the most important for this paper are listed here. But here we were interested in really biodiversity patterns. So floristic patterns of how species composition changes in different habitat types. So here, this is an NMDS plot. So people aren't familiar with these kind of ordination plots, then basically they can interpret it as, so each point represents a forest plot, and then plot points that are close to each other are plots that are similar based on their species composition, and points that are far apart are different based on their species composition. So we can see that there's these kind of quite clear patterns of clustering, which you can see in the different colors. So the different colors are clustered together, and they correspond to these different broad habitat types, these different kinds of forest. And this data set, there's about 2,000 species in this data set. So it's a really diverse region of the Amazon. But if we take that data set and then we subsample it, so we just look at the 99 hyperdominant species that together account for 50% of all individuals, we see basically the same patterns. And then if we do the kind of reverse of that, and we look at the 1900 rarer species, the patterns begin to degrade, and actually they degrade quite quickly the more we look at rarer species. And the point we made here is that actually most of our understanding of beta diversity, of broad scale, regional scale biodiversity patterns, is driven by common species. Because rare species just occur so infrequently that we can say very little about how they change through space, how their composition changes through space, or how they're going to change through time. So rare species in tropical forests that just occur so infrequently that it's really hard to say anything at the moment, at least, about how they change through space and time in terms of their abundances. For all of these studies, there were some significant things missing. So an important one was that they didn't include any small trees. So they started at 10 centimeters was the cutoff. So often in Amazonian forest ecology, we have 1 hectare plots that have a 10 centimeter cutoff. And that excludes lots and lots of trees and lots of species. So some of the most diverse families and genera in Amazonian forests rarely reach 10 centimeters. So lots of Melastomataceae, Rubiaceae, Mataceae, these really important families in Amazonian forests. Many of their species are concentrated in, they're small, they just occur in these small size classes that might not often reach 10 centimeters, so excluding lots of important information. And secondly, there was no size stratified framework, and I'll talk more about this in the next couple of slides. But basically, what I'm talking about here is in all these previous studies, we treat everything above 10 centimeters as the same. Okay, so it doesn't matter if you're very big or very small, it counts as an individual. And then the third aspect that was missing was really this evolutionary context, and I'll discuss that again further in the next couple of slides. So big trees and small trees. So Amazonian forests or all tropical forests have these massive trees, which you know I'm a big fan of. They're good for posing for photos, and they are really important for their role in lots of different ways in the ecology of these forests and in storing lots of carbon, for example. But not all tropical forests look like this. So this image here is from a pecan pole forest in Peru, in the northwest Amazon, so in Loretta. And in these forests, very few individuals will get above 10 centimeters. So if you only looked at trees above 10 centimeters, you'd exclude 95% of the individuals in this forest. So maybe in 1 hectare, 50 individuals about got above 10 centimeters. And this is a really unusual forest, but there's others like this. So white sand forests, for example, equally have very stunted trees. So not only are you excluding these important families and genera that are mainly represented by small stem species, but you're getting a poor representation of many different kinds of forests and different ecosystems that exist within Amazonia. And then there's this idea of not breaking things down, not having a size stratified sampling process. So in this paper from 2016 by Farrier et al., they showed that at least in BCI in Panama, there's this power law relationship between stem diameter and the number of individuals. And basically this shows that you have lots of small individuals in tropical forests, but you have very few large individuals. And this matters then for when we're using these abundance estimates to look at dominance. So really, if we just use this 10 centimeter cutoff, which is indicated by this red line, and consider this all together, we're getting several orders of magnitude more smaller individuals in the 10 to 20 centimeter size class, say, than the very large individuals. So we're studying dominance, then really we're getting an understanding of what's dominating those smaller size classes, rather than what's dominating in the canopy or emergent strata, which are represented by these very large trees. So we have lots of small trees, few big trees, and I think that really matters for how we understand these ideas of dominance and rarity. And the next point then that I wanted that was missing or we wanted to include is this kind of evolutionary context. So we know that some plant families contain lots of common species, and some plant families contain several hyperdominant species. So for in Amazonia, in the Western Amazon particularly, there's lots of palm species. So this was in the original hyperdominance paper led by Hans in 2013. Also, another example would be legumes for Fabaceae, a really abundant family, but lots of dominant species. So it seemed then that these hyperdominants might be clustered in specific lineages. And then in a subsequent paper led by Kyle Dexter, in Drumshav, they showed that species abundance does have a phylogenetic signal. So basically, how abundant you are is dependent on the lineage that you belong to, which families or genera. So specific lineages have very abundant species, and others have very rare species. So in some ways, this isn't new information for botanists that are experienced working in tropical forests, but we wanted to try and quantify then this idea, the idea of phylogeny, of how important it is, what lineage you belong to in terms of the chance of you having a number of hyperdominant species. So then these are the kind of three broad research questions that we addressed in this study. So is hyperdominance persistent across Amazonian tree strata? And then does the identity of these hyperdominant species vary across strata? And then finally, this idea, are hyperdominant species concentrated in specific lineages, and how does this vary across strata? So to answer this question, we needed some new data. So previously, this ATDN, the Amazonian Tree Diversity Network data, consists of plots, these 1 hectare plots with trees above 10 centimeters. Similarly, the rainfall network as well consists of has this 10 centimeter cutoff, but we wanted to look smaller than that. So we looked to the this data set that was established by RedGentry, who was really the master botanist and ecologist of Amazonia. So prior to his death, I think it was in 1990, he established these series of 0.1 hectare, so small plots, across the world really, but heavily concentrated in South America, in Amazonian forests. And in these plots, he identified and measured all stems that were greater than 2.5 centimeters. He also wrote this book, which I'm sure many people are familiar with. It's an incredible field guide to all the woody plants of Northwest South America. But since these kinds of plots were established and he published this work, many people, including myself and all the collaborators in this work, had been establishing these kinds of forest plots where they included these small stemmed individuals. So in this study, we wanted to try and bring this together in a network of plots, and that's what we did. So this is really a collaborative process. So there's 140 collaborators working, contributing this field data towards this study. So I can't list them all here, but Chris Barilotto, who I really established this idea with, was really important, and Flavia Costa at INPA in Manaus, and also Oliver Phillips in Leeds as well, who had established many of these plots. So this really represents decades of people's work, years and years of field work, collecting this really valuable data. And in the end, we had over 1,000 of these small plots, mostly 0.1 hectares, and many of these are subplots then within larger plots. And they represent really important contributions from big networks as well. So the Pepibio network in Brazil, the Medidi network in Bolivia. But together we have this really powerful data set that allows us to kind of synthesize these ideas of tree dominance across these very large scales in Amazonian forests. So in terms of the approach that we took, well, the first thing that has proved a little controversial in some papers recently is just defining what is Amazonia. So to do that, we just used elevation, precipitation, and tree cover data to define this boundary then for the border of Amazonian forests. We also realized that we had this RedGentry network, but because, as I said earlier, those large trees are they're uncommon. We need to add some rainfall plots. So these are 1 hectare plots. So very kindly, members of the rainfall network contributed these larger plots so we could get a better idea of the composition of large trees. And then we basically divided the basin up into these 5 regions, which is signaled by these solid white lines, and 6 strata, which we use diameter size classes to divide that up. So the smallest was 2.5 to 5 centimeters, and that moved through to the largest size classes, which was trees above 50 centimeters. And instead of doing kind of modeling and interpolations like the 2013 Hyperdominance paper, we just used a spatially stratified resampling approach to try and account for the fact that these plots are not equally distributed. So we have much fewer plots in southern Amazonia, for example, in comparison to the Northwest Amazon or Southwest Amazon. So we divided it up into these sampling units, these regions, and then we split them in half, these dashed lines, and then sampled an equal number of plots to try and use all the data that we had, but not bias it towards particular regions. And the product of this work then is that we have abundance estimates for every species in our data set that we think is a fair representation then of what their abundance is across the whole basin. Okay. Now, the next thing I just want to discuss briefly is trying to curate and bring together this kind of floristic data at scale is a massive challenge. So it's over 1,000 plots from different sources. I'd like to make a bit of a plug for forestplots.net, which I think is an amazing resource. So if people aren't familiar with this, this is an online repository for storing this kind of plot data, this kind of floristic data. It's run through Leeds, but really it's not you're not contributing to a network particularly, you're not necessarily sharing data. It's a platform that you can upload your data to, and if you want to share it with other people, you can do that very easily. But also it's just a long-term permanent repository, and it actually made it much easier for the data that was already stored in forest plots. Once people have agreed to share that data, it can be shared at the click of a button, rather than the very long process of trying to harmonize or bring together these different data sets in different formats. The second thing that we did was remove all morphospecies, so we can only use things that have a valid species name. So this really led to tens of thousands of individuals not being used. So there was about nearly half a million individual trees in this data set, but many, we can't use that information because they have, they're just given a morphospecies identification. They're not identified to species level. The next step then is standardizing species names, and another plug then for the taxonomic name resolution service. It does this automatically to remove synonyms. Massively helpful tool, but also this is standardizing names, but it's not really standardizing the taxonomy, and there's lots of issues with the actual species identifications that can't be corrected at this stage. I'll talk about that a little bit more in the discussion about the issues associated there. And then finally, we tried to check all of our species names against expert verified checklists. So they've been produced in recent years. And again, there's been some controversy, but we wanted to make sure that all of our species were well identified, or at least were valid species. Anything that wasn't listed in these kind of expert verified checklists, I went through and checked manually and made sure that that was a valid species by comparing with previous collections. So there was lots of shrubs, for example, that wouldn't have been valid in these lists, but we wanted to include because the kind of decision of what's a shrub and what's a tree is very vague. Anyway, so then once we've gone through this process, then we can start looking at these abundance estimates and really if this kind of phenomenon of hyperdominance, how that works across these different strata. So we wanted to address then this first question of, is hyperdominance persistent across these different strata? And that's what this data shows. So here we have the 6 different size classes on the x-axis, and then we have the proportion of species that you needed to make up 50% of individuals, so the percentage that hyperdominant species accounted for. So it's a bit counterintuitive, but the lower this number, the more dominance there is in that forest. Okay, so for example, this is the Southwest Amazon. So here, about 3% of species in the smallest size classes accounted for half of all individual trees. So in general, we do see this kind of dominance, this hyperdominance across these different strata, but things vary depending on what size class you're looking at, and also across regions. So a whole Amazon basin scale, there's relatively little variation, it's quite consistent. But within different regions, we see the Southwest has a high level of dominance, whereas the Guiana Shield has much less, particularly in small size classes, but that increases if you look in larger size trees. In general, you see less dominance in these large canopy and emergent strata. So this point then that the Southwest Amazon is particularly, it has a high level of dominance is relevant. It's interesting because this original work that was published by Nigel Pittman was really initially based in much of these forests in the Southwest Amazon. So it seems that those forests in particular have a high degree of dominance, but broadly it's shared across Amazonian regions. Now this number, this percentage of species that you require to reach hyperdominance is not driven just by the common species, but also by the rare species. So the more rare species you have, the easier it is to achieve a small percentage, if that makes sense. So we wanted to test whether this differences among regions is just driven by differences in species richness. And actually, to some extent, if you look at the whole Amazon basin, you see where you have lots and lots of species at a whole Amazon scale, then you do get this high degree of dominance. But if you look within the different regions, there's no pattern there. There's very little pattern, which suggests that actually there is differences among regions. So some regions are more dominated than others, and that's not entirely driven by the species richness, the number of rare species. Oh, sorry, skip the slide. So the next question that we wanted to address was, does the identity of hyperdominant species vary across Amazonian tree strata? So here we have, this is just a kind of a two-dimensional histogram that shows the number of hyperdominant species that occur in 1, 2, 3, 4, 5, or 6 different size classes along the x-axis, and then the y-axis is the number of hyperdominant species that are hyperdominant in 1, 2, 3, 4, or 5 regions. Now the point here is that most of these hyperdominant species are quite specific. So they only achieve dominance in one region or one size class. There's very few that achieve hyperdominance in many size classes and many regions. And there's only one species which was hyperdominant across all size classes and across all regions, which is Escherichia coriacea. And I'll talk a bit more about this species later and issues associated with its identification and whether this really is one species. We can also summarize these patterns of hyperdominant species composition then with these ordination graphs. So this is another NMDS, which shows, again, it's a summary of species composition across these different strata. So this is just hyperdominant species across the 6 different size classes, 6 different strata from the smallest. So these are small understory trees to the largest rings, which are the large emergent and canopy trees. And then the colors again correspond to the different regions. And you can see there's clear patterns. So species, these hyperdominant species are clearly, they vary across this x-axis in terms of the different regions, the different colors. And then there's this second compositional axis of size class. So they vary across size class and they vary across region. And that's very clear, I think. So different species dominate the understory compared to those species that dominate the canopy. And that varies across regions. So the next question then that we wanted to address was, are hyperdominant species concentrated in specific lineages? And how does this vary across forest strata? So there's, again, another graph with lots of information here, but this is just an example of these 3 plots are from different strata. So from the very, very understory, the small trees, and the 2.5 to 5 centimeters, then somewhere in the middle, the 10 to 20, and then above 50. And then each point here represents a family. And the question that we're asking here is the richness of that family, does that dictate the number of hyperdominant species that we find within it? So families that have lots and lots of species, do they also have lots of hyperdominant species? That makes sense. So you have a very species-rich family, would we expect that to dictate the number of hyperdominant species within that family? Okay, so in the smallest size class, we see, sorry, in this 1-to-1 line, this white line represents what we'd find if that was the case. So if it was purely dictated by the number of species in that family, all families would line up along this white line, but we see some significant deviations from that. So some examples then, if we look in the understory, a number of families are more abundant, or they have more hyperdominant species than we'd expect by chance. And these include things that are, people that have worked with botanical data in Amazonian forests would recognize. We see lots of Burseraceae, so lots of proteums, which are very dominant. Ericaceae, palms, are also there. Violaceae, also really well represented, or have more hyperdominant species than we'd expect by chance. The flip side of that is that we see these families that have lots of species, but very few dominant species. So again, this will be kind of familiar to people that have experience working in Amazonian forests, but lots of Rubiaceae in this instance, Lauraceae, and Melastomataceae, really diverse groups, but no or few hyperdominant species. And as we move through size classes, this kind of changes. So we see different things emerging. So particularly in these mid-size classes, we see the onset of Moraceae, so it's Ficus and Brosimum, less dominant in the understory, more dominant in these mid-story size classes. And then by the time we get to the emergent canopy, most species kind of line up with our expectations. So for Fabaceae, so these are legumes, really, really important, by far the most species-rich family in Amazonian forests. And it's only actually in this emergent canopy strata above 50 centimeters in diameter that they have more dominant species than we'd expect by chance. Now we can look at these kind of relationships more formally. So in this phylogenetic analyses, we basically plotted these hyperdominant species onto the angiosperm phylogeny. So this is a complicated figure, looks a bit like the Death Star, but basically you can interpret this. So each tip that is colored is a genus that contains at least one hyperdominant species. Any tip that has a name, that's a genus that has at least 3 hyperdominant species. And then the color corresponds to the size class which those hyperdominant species correspond to. So for example, pepper genus, it's blue, so it has at least 3 hyperdominant species, but in these very small size classes, which makes sense because this is a really important understory genus. So I hope that's clear. So this is a genus level phylogeny that was developed by a collaborator on this project, Fernando Coelho, who published this in Nature, Ecology and Evolution in 2019. But the broad pattern then that we can see from this figure is that hyperdominant species distributed across the angiosperm phylogeny. It's very hard to see, at least for me, any strong patterns. We see some kind of gaps in Rubiaceae maybe, but it's hard to know if there's any real structure here. But what we can do is we can start to get some metrics from this. So what we used was the mean pairwise phylogenetic distance. So what we did then, we take 2 genera and we measure the distance. So this is the length of all these nodes on the tree. We measure how far that is, and then we do that for all the hyperdominant species in each size class in each region. And that's what this next figure shows. So we take all those pairwise phylogenetic distance for each size class in each region. So size classes along the x-axis, region is again labeled with the color. And then we compare that then to the kind of phylogenetic distances that we'd expect if we sampled them randomly across the phylogeny, which is the white line. This is our null expectation. Again, it's quite a complicated figure, but basically you can interpret things as points that are above the line are more dispersed across the phylogeny than we'd expect by chance. And then points that are below the line represent groups of hyperdominant species in a particular size class in a particular region that are more clustered than we'd expect by chance. And we see this contrasting pattern across size classes. So hyperdominant species that are dominating in the 2.5 to 5 centimeters are consistently more dispersed across the angiosperm phylogeny than we'd expect by chance. And the opposite is true of these very large stem species. So if you're above 50 centimeters, they tend to be more clustered than we'd expect by chance. And we see that, we can see that in the data. So in these large size classes, there's lots of hyperdominant species in specific families and specific genera. So there's many Fabaceae, lots of legumes, lots of things in Licitidaceae, Eschweileras or the Brazil nut. So that pans out when we just in our observations of the data as well. But the pattern is clear. We can also use then these mean pairwise phylogenetic distances to look at, again, at these ideas of composition, but instead of basing our NMDS figures on the abundance of these hyperdominant species. We can do that based on the evolutionary relationships. So this figure is analogous to this one that I showed before. So the big circles show the hyperdominant species in the large size classes, the small in the small understory size classes, and again, color corresponds to region. And the point that I think this figure makes is that species that are hyperdominant in large size classes, they're very similar across regions. They come from similar genera, similar families. So just a few families in genera, it's the same kind of idea. They're dominating the canopy and emergent strata, and they're doing that across the basin in different regions. Alternatively, stuff that's dominating in these small size classes is very different in different regions. They come from different lineages in these different areas of the Amazon, these different regions. Okay, so then I'd like to just move on and think about what the implications are for these results. So we find that it really matters where you are in the canopy in terms of your identity as a hyperdominant species. So species that dominate in the understory, very different to those that dominate in the canopy. So then the next big question is why? What allows these specific lineages to be dominant? What characteristics do they have? What functional traits do they have? And I think for me, that's the big next question. What is it that allows some of these species to exist at very high densities, at very large scales across Amazonia? And for that, I think we need some detailed functional trait analysis. So I'm particularly interested in thinking about these species in terms of Janssen-Connell hypothesis, negative density dependence, what allows them to form dense stands, but also what allows them to be dominant at very large scales. And I think some understanding of their dispersal and dispersal traits would be really interesting in that context. The other big result that I showed is that we have these very contrasting phylogenetic patterns. So these understory hyperdominant species that are dispersed across the phylogeny, whereas the large stem species are clustered. They're phylogenetically clustered, these large hyperdominant species. So what has led to this distinct pattern? Well, the fact that they're phylogenetically clustered, the large stemmed individuals, I think suggests that these species have been, firstly, that only a few lineages can make it to the canopy to those emergent strata, can really grow that big. But the species and the lineages that have developed that ability to grow big have dispersed and been very successful at very large scales. So just a few things achieve dominance in these very large, the upper strata of the forest, but then they've dispersed widely at very large scales. Then the dispersed hyperdominant species in the understory suggest to me that maybe there's they've been less dispersed across through evolutionary time, so they've been more isolated. So different species and lineages have evolved to dominate in different regions of the Amazon, and there hasn't been much dispersal among them. So there's been some isolation there. That geographic signal that we see also may suggest that there's different kinds of environments in different parts of the Amazon, and we know that there's structural variation across the Amazon, which is fairly consistent. So forests in eastern Amazonia tend to be very tall and less dynamic, so there's less turnover in species, there's less gap dynamics. In the west, the forest is shorter, but it also is much more dynamic, there's more gaps, there's more light in the understory. So maybe certain lineages have evolved to become dominant in that more open understory, whereas in the Guiana Shield, where it's more of a closed canopy, less dynamic, a different set of species have evolved to become dominant there. Another really important implication, I think, of this research is about these large trees that are phylogenetically clustered in just a few lineages. So they lack phylogenetic diversity. And does this lack of phylogenetic diversity mean that they could be more vulnerable to, for example, climate change? It's also important to recognize that it's these big trees that are really driving a lot of the carbon cycling. And it's also these big trees that will be exposed, I think, to the kind of most brutal changes in climate. So they're not buffered by the canopy. They exist in hot, dry environments above the canopy, which provides some climate buffer. So are they more vulnerable then? And does the fact that there's just a few genera and families that really dominate those strata, does that mean that there's potentially less functional variation there to deal with that the changing environment that they're exposed to? So another important point is this single species that was dominant across all size classes in all regions, Eschweiler coriacea. So it's probably the most common species across the entire Amazon, but it's also hard to identify. So it's a difficult species to identify, and this is not limited to this particular species individual, but many species in Amazonia are really difficult to identify using traditional morphological approaches. And not only are they hard to identify, it's really hard to standardize identifications across these kinds of synthetic networks. So we have lots of different groups working in lots of different regions. So in that instance, it becomes a major challenge then. Are we all calling the same things by the same names? Are we all identifying the same species consistently? And where we've tested this question, possibly not. And I think there's some emerging research that is focused on this one species, the most common species, that suggests it might well be a species complex that actually consists of a number of discrete species within the Eschweiler coriacea complex. Another example of that came out very recently in the literature, just a couple of weeks ago, it's Proteum heptaphyllum. So the Proteum is a really important genus in Amazonia, and it's the genus with the greatest number of hyperdominant species. So there's lots of hyperdominant species, and Proteum heptaphyllum is one of the most common. But a recent study that looked at the genetics, also the morphology and the traits of this species across the Amazon basin suggests that it may actually represent 8 distinct species. So this kind of questions much of this concept of hyperdominance. These common species that we see across the basin, some of them at least appear to be species complexes. So I think there's some really important questions that we need to answer about being able to identify species consistently in these synthetic large-scale data sets. And really, does it matter? So in the area of functional ecology, is it still important to identify and understand species? So for me, my opinion is it really does. I think that much of the changes we'll see in Amazonian forest vegetation will be determined by species. And maybe if we could measure all functional traits, across species at very large scales, it becomes less relevant, but we can't do that. And I think that for me, species and the species concept is vitally important, and we really need to address these issues of taxonomy. So I recently led a review that kind of addressed these issues of how do we deal with these issues of identifying species in tropical forests, especially when we don't have fruit or flowers to identify them. So about 10 years ago, or people would have suggested that this would have been revolutionized by DNA barcoding. But I think subsequently we've seen that doesn't really work well in these hyperdiverse Amazonian forests. And I would make I'm a proponent of using spectroscopy. So this is leaf level spectroscopy, where we're measuring reflected light from leaf surfaces. And we can use this approach to identify species based on their reflectance signal. And crucially, we've got a lot of existing resources. So we have these herbarium samples. We have permanent forest plots that we can use to draw on and develop spectral libraries, and then use new tools from artificial intelligence, deep learning networks to come up with quantitative species identifications that are repeatable quantifiable and also linked to or underpinned by next generation sequencing. Also, this kind of spectroscopy approach links to remote sensing, and we're able to measure some similar kinds of attributes from above, from drone or plane platforms. And it also tells us a lot more than just the identity of the species. So it tells us a lot about the functional traits of a given species. So not just what species it is, but about the leaf chemistry, about how it works. So for me, I think that is a really exciting way forward. I'm very keen to pursue. And then finally, on this issue of remote sensing, then I think this approach has really important implications for remote sensing. So traditionally, we see the forest something like this. Now we see the stems, but in fact, now we're viewing forests and trying to measure biodiversity more and more from above, from planes, from drones, from satellites. But actually, we're really just seeing a fraction then of the composition of a forest, even in terms of the dominant species. The dominant species we've shown in this paper are very different from those that are dominating the canopy. So we end up, instead of seeing the forest on the ground, we have an image like this. So this is taken from a drone, and we have here about 2 centimeter resolution, and we can see lots of detail here and lots of tree crowns, but it's still just a fraction of the species that are dominating this system. And then if we think about using satellite platforms or plane platforms, this pixel size increases from 2 centimeters to maybe 2 meters with a plane, and then up to 20 meters with a satellite. So then we're really just seeing the forest that's going to be driven by these large stemmed individuals, and they're going to be driven by those dominant species that we've identified in this paper. So just a few hundred species really then are driving that signal that we're getting in remote sensing. So this is an issue that I think we need to spend more time quantifying. But I think there's some good news. So in another previous study, we looked at how species composition varies across landscapes in Peru. So this is in Apuamshana, near to Iquitos. And on the left here is a true color image. This is taken from the Global Airborne Observatory, which is an imaging spectrometer on a plane. And then the central image here shows variation in the spectral signature, then the spectral reflectance of this canopy, this forest canopy. So here, light colors are similar to one another based on their spectral reflectance. Dark colors are similar to one another based on their reflectance. But importantly, across this landscape, we have lots of field plots, lots of field data. And many of these are these kind of gentry style plots, these 0.1 hectare plots that are really, most of the individuals in the plots are small. They're smaller than 10 centimeters, but also there's trees that we won't see from above in the canopy. But what we found then across this landscape is the spectral similarity that we get from the spectrometer on the plane. If we do that for the locations where these plots are, and we compare that to the floristic similarity, which is mainly made up of understory trees, you get this very tight relationship, which suggests that although the composition of species might be different in the understory and the canopy, there's some covariation there among them. So with that, then I'd just like to summarize what I've talked about today, which is that hyperdominant species composition really varies across strata and across regions in Amazonia. But there is some covariation then between hyperdominant species across strata. So this makes sense, I think, if you think about an edaphic gradient, the canopy species and the understory species are different, but as they cross that edaphic gradient, they change together, they change at the same time. There's an important covariation. There's also very different phylogenetic patterns in these hyperdominant species across strata. So understory species belong to lineages from across the angiosperm phylogeny, whilst canopy and emergent hyperdominant species are concentrated in just a few key lineages. I think this framework is useful. I think it provides a tractable pathway for then understanding half of Amazonian forests in the near term. So it's near-term solutions for quantifying biodiversity, but really provides this insight into how things vary depending on what strata of the forest you're interested in. and it has important implications for how we interpret remotely sensed forest imagery. But I think there's some really large and important questions about the underlying taxonomy of dominant species. And here we're talking about dominant species, but those figures I showed earlier, what about these very rare species where we have 10 individuals? How can we be really sure of those that is a valid species name? So I think there's lots of work to be done on taxonomy and working with systematists, but also integrating these new technological approaches. Finally, I'd just like to thank, this is a really hugely collaborative project, and I felt really grateful that all of these different researchers from across Amazonia shared this really valuable field data. So I'd like to thank them for their contribution, because I understand how hard it is to collect that data. And then I've been based at these different institutions doing this work, and it was funded mainly through my Marie Curie Fellowship. And then with that, I'd take any questions, but feel free to email me or tweet me. And this is the paper that was published this year in Nature, Ecology and Evolution. So thanks very much.
Thank you, Freddie. That was terrific. Really, really nice overview. If you'd like to stop sharing your screen,
okay, yeah,
I'll open up to questions. And any of you feel happy to switch on your cameras, feel free to be a visible face in the audience. Okay, and so either just pop a question in the chat or raise your hands, and I can direct them to Freddie. I'll kick off while we're waiting for the questions to pop in. So the key question is around what creates this hyperdominance, and you're familiar with Carolina Levis's paper and argument that there's a indigenous footprint that on what's hyperdominant. So, and what has your insights into the lower strata? How does that inform that argument? Has it supported her argument, or does it actually suggest there's more of a random, non-human factor there?
Yeah, it's really, really interesting. It's something we discuss in the paper actually, and I think what our data shows a little is that many of these kind of domesticated or trees that we believe, or people believe, were really important, particularly to pre-Columbian communities, peoples in Amazonia, are hyperdominant species, but mainly in the smaller size classes. So lots of those species that have been identified belong to these smaller size classes only. And at least the idea that we put forward in the paper is that it's kind of easier to manage or manipulate or propagate these smaller species rather than trying to do that for very large trees. It's definitely not the case in all. There's many cases where large trees have been really important. So the Brazil nut is a really good example of a massive tree that we think has been really important. But I think to me it seems like a logical argument that many of these common understory species are those that might have been important to people over long terms in the Amazon, and that has probably affected their abundance in a big way.
Mm-hmm. Okay. Any more questions? You shy? I have a few more, but feel free to ask them. So in terms of this pattern of hyperdominance, I'm trying to think, well, obviously these are open questions about what drives that. And you mentioned that we were going to discuss historical legacies. You mentioned the possibility of traits determining them. And to what extent do you think hyperdominance could essentially be a random process as well, where over time certain species just sort of increase to critical abundance, and the more that they are, the more they push towards hyperabundance, and over time, is there a cyclicity of these? I'm not sure you have the answer to this. I'm just speculating whether, to what extent there's randomness in this process. And clearly you're seeing some phylogenetic signal that suggests that it isn't entirely random. So there's a non-random process as well.
Yeah, I think it's a really good question. So I don't have a solid answer for I'm not sure exactly what our expectation would be, based on if this was entirely random processes, but I think we wouldn't expect these species to achieve such high abundances. But yeah, it could be interesting to think about some theoretical expectations about what we might expect, and see how the observations match up. I think there's a couple of things that suggest it's probably not random. So one is the phylogenetic signal, but also these kind of longer term records, maybe these historical records are looking, if you had some paleoecological records, for example, particularly of palms or things like that, have their abundances, have they always been so abundant? And I think, off the top of my head, I think some of the key palm species like Mauritia flexuosa, Iriotea deltoidea, these really super common palm species have been abundant for a long time. So if it was random, I think you'd expect a bit more variation.
Mm-hmm. Andy?
Hi there, great talk. I enjoyed that. I'm sorry if this question, you already answered it at the beginning of your talk. It took me a while to dig the link out and I was a little late joining. So apologies if you've already dealt with this. But so this idea of the size classes, and that's really interesting that the patterns differ in different size classes. And I guess what's important there, one of the things that I was wondering about is obviously the species might differ in sizes, right? And so there may be some species that are restricted, say, to the lower size classes, whereas you've got other species that might, the largest ones obviously can range across the whole range. The adults will be the largest sizes, but younger individuals will be in the smaller size classes. And so you could be seeing two patterns. That what you're seeing could be the result of two processes, I guess, right? One is the abundance of species with different maximum sizes, and the other is changes in abundance as species grow through these size classes. Do you see what I mean?
No, I see exactly what you mean. Yes, so in the understory, you have a mix of things that are going to get big and things that are never going to get big, and yeah, you're kind of seeing those two things together. And I don't think I have a very strong answer for how to disentangle that at the moment, or I don't think we really fully address that in the paper. So yeah, I mean, just looking at the pure abundances, some of those species we know that are hyperdominant in the understory grow to be very big. So Escherichia curiaceae is a good example. So yeah, you're right that you do have in the smaller size classes a mix of things that dominate, that are specialists there in the understory, and then things that are go through to the canopy. I think for the phylogenetic patterns, I think actually I would expect the signal to be stronger if you look just in the understory. I think it's those understory things that are kind of come from all over. So the pipers and I don't know, the Eugenia and the Metaceae, all these different distant lineages, the Rubiaceae, things that are underrepresented are often these understory, more specialist understory species.
Thank you. It'd be really interesting to see how those two things come out as you tease them apart. Thank you.
More questions? You were saying, Freddie, about using hyperspectral signals as a way of trying to distinguish species. Is that, and how feasible is that for species that are so similar looking, at least in visual wavelengths? Do you think there is enough information?
Yeah, I mean, I think it's so there's a couple of things. I think at the leaf level, I think if you're looking with a spectrometer at a leaf, so this is not remote sensing from above the canopy, you're just looking right there at the leaf. I think there's lots of evidence that it's a really it's a really good tool for identifying species, that you can see species have different spectral reflectance properties, and I think that's driven by defense compounds really. So there's a few papers, one Paul Fine led a paper this year that with Greg Asner, which showed that in Proteum, so a really diverse, important genus, you can see these very fine scale spectral differences, and that corresponds very nicely with, if you do the metabolomics then on those individuals, you see those differences. So I think at the leaf level you can. I think it's an open question, and it's something that I'm exploring or would like to explore more. I think it's going to depend on which family or which genus. I think it's going to depend on which group you're looking at, how effective it is. But there's also a lot of work coming out of Beto Vincentini in Brazil, who's using these approaches across lineages, and it seems to work really well. I think when you start trying to use hyperspectral imagery, so you're looking above the canopy, it becomes then much more complicated, because you're not just looking at the reflectance of the leaf surface, but you have all the structural parameters as well. So again, I think it's an open question. I think there's some evidence that it does work well for certain species that have big crowns that are really visible, dense crowns where you're seeing a lot of the leaf surface. But how well that's going to work across lots of species, I think we don't know. I think it'd be great if you could, mount a really high quality imaging spectrometer on a drone, where you have this very fine spatial resolution and you can isolate the leaf surface of crowns. But yeah, it's an exciting area of research, but yeah, lots of uncertainties. But I think it works well.
Okay, Pierre-Michel?
Yeah. Well, thank you, Freddie, for your presentation, and it's impressive to see all this data. I have a question which is related to the seed dispersal, of course, of all those species. When we look at your phylogeny, it's obvious that many of the large trees are wind dispersed, and many of the understory plants and these low strata are either explosive or dispersed by small birds. And how do you integrate that? What is the effect of fruit seed size and dispersal mode of dispersal in your analysis?
Yeah, so I guess it's something we haven't considered formally at the moment. I think that this is the kind of explanation I think would really help understand why the patterns are so different in these different in the different strata. So I think, yeah, if the stuff that was dominating in large size classes was all wind dispersed, then and the stuff that was dominating smaller size classes was more dependent on small birds, for example, then that might provide a kind of mechanistic explanation for why we see these phylogenetic patterns. But I don't have that data on the different dispersal modes of those species to really get a good handle on that. But I think it'd be super helpful to understand that better. I think it's really, really crucial for these kinds of mechanistic understanding of dominance. Yeah.
I imagine for the hyperdominance, it wouldn't take too much effort to try and get the dispersal syndrome for those. It's quite interesting to see if that has an analysis of that.
Yeah.
That holds.
If nobody is asking, can
Go
I
ahead, yeah.
just develop a little bit more? Yes, there's this very interesting paper by Lohmann and Connell in '89, and the other paper by Hart and Hart in '89, same time, in American Naturalist, about the importance of large seeded seeds being dominant, and so it's more or less related to that. I think we should have more interest in the type of fruits and seeds to explain which species will be dominating or not dominating. And another point that is barely considered, I think, is the impact of human beings in the last centuries and millenaries. And it's clear that many of those seeds have been transported by humans over long distances. Just cited recently a paper by Darrell Posey in just mentioning that many of the island forests in Para could have were in fact totally replanted by human beings, like Kayapo Amerindians. And we know more or less now that a large part of the Amazon has been entirely manufactured by human beings. So what we are observing today is maybe simply the consequences of long-term use of the forest by human beings, and the trees are still there because they have a long lifetime. And I remember being in Suriname and seeing a big grove of Astrocarium said to be Theophyllum in this time, but it's Pharenosum now. And it was clearly due to human beings. But if you're not considering human as an agent of dispersal, then you're missing the story, in fact. So I think it is something that we botanists and ecologists have to reconsider more and more frequently in these days.
Yeah, I completely agree. I think that, yeah, I mean, we touch a bit on these themes in the paper about the impact of humans on these species, but I think it's a hard thing to quantify, and I think we don't include it enough. But I think it would really help in these explanations, and like Yudvinda mentioned initially, how we may expect this to vary across size classes as well, I think is interesting as well. If these humans have had different influences on dominant species in different strata of the forest. So, but yeah, I'd love to see more and be involved in thinking about this more.
Just to alimentate the discussion.
Yeah. Well, I think your last point about Amazonia being a manufactured system, I think that some people that would disagree with you, as Mark Bush, in a few weeks' time in his seminar, I think would present a counter opinion on that one. But I think it's overall, it's certainly an interesting question to look at the legacies of past human use and shaping the forest composition and dominance. Okay, any more questions? If not, go ahead.
Yeah, I thank you for your talk. Fantastic to look at the understory or smaller trees in the Amazon forest. And I have a question. You showed us a pattern of a geographic pattern of southwestern, northwestern Amazon. The hyperdominance of the understory is higher there. Did you look at the forest turnover rate?
No, we didn't. We didn't have, in terms of through time. So how species turns over through time, is that? And no, we didn't include that information.
Yeah, it looks like it's similar pattern of the forest turnover rates of the Western Amazonia is higher than, and so I was wondering if the role of disturbance in this pattern.
Yeah, I think it's really important, particularly in the Western Amazon, where you do have this more open, more disturbed, a more open understory. You have much higher light. So yeah, you have this more distinctive understory there. The composition is more distinct because it has more light compared with the east. So I think that pattern plays out. I think that is, we see that difference, that difference in composition.
Thank
you. Okay, thank you. If no more questions, I suggest in seminar tradition, everybody unmutes their microphone and we give an audible round of applause to Freddie. Thank you for your work and good luck with the future directions of this. This is a fascinating topic.
Cheers. Yeah, thanks very much. Yeah, it's a real pleasure to present. Yeah, thank you.
Great. I look forward to seeing new developments and good luck with this network as well. It's fascinating to have really pulled together this RedGentry network of small trees. I think it's a really, really valuable resource for the community.
Yeah, I hope so. And I really love people especially the people that contributed data to take on new projects using this data set. So yeah, that'd be great. That'd be a great result if it became widely used. So yeah. Thanks.
Great. Thanks, Freddie. Thank you, everyone.
Cheers.
See you
Bye-bye.
next week.